Hello students, in last video we started a topic and the name of the topic was cilia and flagella, and there we discussed a difference between cilia and flagella, and we said that cilia are small in size and flagella are large in size. All cilia work together, whereas flagella always work or beat independently. We also said that cilia are mostly found in eukaryotic cell, whereas flagella is found in both eukaryotic and prokaryotic cell. And cilia are useful in locomotion, food gathering and sensing of the environment, whereas flagella is useful in locomotion. And we said that cilia are many in number and flagella are few in number. And cilia is found in paramecium and flagella is found in bacteria and sperm. And today we are going to discuss about cilia and flagella in detail. Okay, so let's learn about cilia and flagella in detail. So the first point that we will say about cilia and flagella is that they arise from basal body. They arise from basal body. Both cilia and flagella arise from the basal body. So we are learning cilia and flagella. Cilia and flagella. And the first point in this topic is they arise from basal body. And this basal body is situated inside the cell membrane. It is situated inside the cell membrane. Okay, we are learning cilia and flagella, and in that we will give the first point that they arise from the basal body. Both cilia and flagella arise from what? They arise from basal body, and this basal body is situated inside the cell membrane. This basal body is situated inside the cell membrane, and then we will say that both are covered by cell membrane. Both cilia and flagella are covered by cell membrane. Both the cilia and flagella are covered by cell membrane. Okay, so the first 2 points regarding cilia and flagella are that they arise from the basal body, and this basal body is situated inside the cell membrane, and both of them are covered by cell membrane. These are the two initial points regarding cilia and flagella. Now we will discuss the cross section of cilia and flagella. We will talk about the cross section of the cilia and flagella. For that, we will draw the figure of cross section of the cilia and flagella. Okay, after talking about these 2 points, we will draw the figure of the cilia and flagella. So we are drawing figure of the cilia and flagella, and this figure explains cross section of the cilia and flagella. This figure explains cross section of the cilia and flagella. So we are drawing cross section of cilia and flagella. So we are drawing cross section of the cilia and flagella. Okay, so this is a cross section of cilia and flagella is there, and in this cross section the outermost part is cell membrane. This figure explains cross section of the cilia and flagella. Here we have drawn the cross section of the cilia and flagella, and on the outermost part the cilia and flagella has cell membrane. So we said that cilia and flagella are covered by cell membrane on outer part. Okay, so this outer part represents cell membrane, or it shows cell membrane. So the point is proved. Okay, the first point which we said was that cilia and flagella are covered by cell membrane. So that point is proven here, that this figure has outermost part called cell membrane. Okay, because this is the cross section of cilia and flagella, and the outermost part is cell membrane. Okay, so the first point has been proved. Okay, and now the structure that is inside this cell membrane, is known as axoneme. This structure is known as axoneme. exonym. This is exonym of the cilia and flagella. Okay, this whole structure inside the cell membrane, this whole structure inside the cell membrane is known as exonym, and this exonym has two regions. One is peripheral region and one is central region. This whole structure is exonym. This whole structure inside the cell membrane is exonym, and this exonym has two regions. One is peripheral region and one is central region. Okay, so we are talking about cross section of the cilia and flagella, and in that we said that outermost part is known as cell membrane, and whatever the part that are present inside the cell membrane are known as exonym. Okay, so entire region inside the cell membrane is known as exonym, and this exonym has two regions, peripheral region and central region, or exonym has two parts, peripheral part and central part. Okay, and in the peripheral part of the exonym there are doublets of microtubules which are arranged parallel to each other. Okay, in the peripheral part of the exonym the doublets of microtubules are there which are arranged parallel to each other. Okay, so how many doublets are there? See, this is one doublet, this is second doublet, third, fourth, fifth, sixth, seventh, eighth, and 9 doublets of microtubules are there. These are nothing but microtubules. Microtubules. So, in the peripheral part of the exonym, 9 doublets of microtubules are there which are arranged parallel to each other. In the peripheral part of the exonym, 9 doublets of microtubules are there which are arranged parallel to each other. Okay, so I am repeating the sentence again and again. In the peripheral part of the exonym, 9 doublets of microtubules are there which are arranged parallel to each other. Okay, and all these doublets are attached to one another or each other. They are attached to each other. Say for example, this is the first doublet, and the first doublet is connected to second doublet via linker. Okay, via linker. Okay, so first doublet is second to second. First doublet is attached to second. Second is attached to third. Third is added to fourth. Okay, all are interconnected. All are interconnected, and they are interconnected with the help of what? They are interconnected with the help of linkers. With the help of linkers. Okay, so all doublets are connected to each other via linkers. All doublets are connected to each other via linkers. These are linkers. Okay, these are linkers. Linkers. Okay, so total 9 doublets of microtubules are there. Total 9 doublets of microtubules are there, and these doublets are attached to each other via linkers. Okay, chap. And if this cilia and flagella belongs to prokaryotic cell, if this cilia and flagella belong to prokaryotic cell, generally cilia is not found in prokaryotic cell. So say for example, if this cilia belongs to prokaryotic cell, okay, then in the central part nothing will be there. Okay, if the flagella belongs to prokaryotic cell, then the central part will be empty. Central part will be empty. So here peripheral part has 9 doublets. Peripheral part has how many doublets? 9 doublets. And in the central part there is nothing. In the central part nothing is there, so we will write 0. Okay, so 9 doublets of microtubules are present in the peripheral part, and in the central part nothing is there. So this flagella is prokaryotic flagella. This flagella is known as prokaryotic flagella. Prokaryotic flagella. Okay, prokaryotic flagella has 9 plus 0 type of arrangement. Prokaryotic flagella has 9 plus 0 type of arrangement. Why? Because peripheral part has 9 doublets and the central part has nothing. Central part has nothing. So there is 0. Okay. So prokaryotic flagella has 9 plus 0 type of arrangement. Prokaryotic flagella has which type of arrangement? 9 plus 0 type of arrangement. Why? Because in the central part nothing is there, and peripheral part has 9 doublets of microtubule. Okay, and say for example, this is eukaryotic cilia flagella. Okay, if this is eukaryotic cilia and flagella, then central part has one pair of microtubule. Central part will have one pair of microtubule. Okay, This is one pair of microtubule and they are connected to each other via linker. They are also connected to each other via what? Via linker. Okay, so this is linker which is connecting this one pair of microtubule. This is central microtubule. These are known as central microtubules. They are connected to each other via linker and both of them are covered by central sheath. This is what? This is central sheath. This is central sheath. This is central sheath. Okay. So, say for example, this is eukaryotic cilia and flagella. Okay. Consider that this is eukaryotic cilia and flagella. So, in the central part, it will be having one pair of microtubules and both of them are connected to each other via linkers and both of them are covered by central sheath. So, one pair of microtubule is there, they are covered by central sheath and they are added to each other via linkers. Okay. So, total 9 doublets are there. Total 9 doublets and one pair of microtubule is present in the center. One pair of microtubule is present in center. So, this is known as eukaryotic cilia and flagella. Eukaryotic cilia and flagella. Okay, so eukaryotic cilia and flagella has 9 plus 2 arrangement. Eukaryotic cilia and flagella will have 9 plus 2 arrangement. But prokaryotic flagella will have 9 plus 0 arrangement. Okay, prokaryotic flagella will have which type of arrangement? 9 plus 0 type of arrangement. Eukaryotic cilia and flagella will have which type of arrangement? They will have 9 plus 2 type of arrangement. How can we say that? Because in the central part, one pair of microtubule is there in the eukaryotic cilia and flagella. In the prokaryotic flagella, the central pair of microtubule is absent. In the prokaryotic flagella, the central pair of microtubule is absent, whereas in the eukaryotic cilia and flagella, this central pair of microtubule will be there. Okay, and they are connected to each other via linkers and they are covered by central sheath. They are covered by central sheath and they are connected to each other via linkers. Okay, chalo. Now let's talk about another structure which is known as radial spock. See, these peripheral doublets are attached to central pairs. Peripheral doublets are attached to central pairs via what? Via radial spock. Via radial spock. So this structure is known as radial spock. This structure that I am drawing, this structure which I am drawing is known as radial spock. This is known as radial spock. This is what? This is radial spock. Okay, radial spock. What is radial spock? Radial spock is the structure which connects peripheral microtubule to the central microtubule. Okay, if the central microtubules are present, that will be eukaryotic cilia and flagella. Okay, so in eukaryotic cilia and flagella, the peripheral microtubules are connected to central microtubules. Peripheral microtubules are connected to central microtubules via radial spock. Okay, peripheral microtubules are connected to central microtubules via what? Via radial spock. Okay, so we have discussed the cross section of the cilia and flagella. In that we said that outermost part is known as cell membrane and the structure inside the cell membrane is known as axoneme, and this axoneme has a parallel arrangement of microtubules. This axoneme has parallel arrangement of microtubules and the 9 doublets of microtubules are present in the peripheral part of the axoneme. 9 doublets of the microtubules are arranged in peripheral part of the axoneme. Okay, and all these doublets are attached to each other. All these doublets are attached to each other via linkers. Okay, and if the prokaryotic flagella is there, then the central microtubules will be absent. But if the eukaryotic cilia and flagella is there, then it will have central pair of microtubules. Okay, so central microtubules are present in the eukaryotic cilia and flagella, and central pair of microtubule is absent in the prokaryotic cilia and flagella. Okay, sorry, so it's prokaryotic flagella only because cilia are not found in prokaryotic cell. Okay, so in prokaryotic flagella, central pair of microtubule is absent, but in the eukaryotic cilia and flagella, central pair of microtubule is there. So prokaryotic flagella has 9 plus 0 arrangement, whereas eukaryotic cilia and flagella has 9 plus 2 arrangement. Okay, then these two central microtubules are connected to each other via linker and they are covered by central sheath. And the peripheral doublets are attached to central microtubules. via radial spoke. Okay, peripheral doublets are attached to central microtubules via radial spoke. Okay, so this is all about the structure of cilia and flagella. Please remember that prokaryotic flagella has 9+0 arrangement because central pair of microtubule is absent. But eukaryotic cilia and flagella will have 9+2 arrangement because central pair of microtubule is present. Okay, so this is all about structure of cilia and flagella. Please draw the diagram in your rough note or fair note properly. Then and then you can understand this topic well. Okay, otherwise you can't understand this topic. So to understand this topic, you have to draw the diagram first 2-3 times at least, and then you can read the topic. You can watch the video, you can read the from the textbook also. Then and then only you can understand this topic. Okay, so first draw the diagram, watch the video 2-3 times, and then read it from the textbook. Okay, so it is all about cilia and flagella. In the next video we will learn about centrioles and centrosomes. Best of luck.